Cantwell, welcome to Summit University Forum, and thank you for being our guests. Dr. Strecker, you say that AIDS was predicted, requested, produced, deployed, and now threatens the extinction of mankind because it works. Let's go through this step by step. When and where was AIDS predicted?
Right. We actually believe that the AIDS virus was predicted as far back as 1972, perhaps even earlier, as far back as in the early 60s. Writing in, and I will address that by first turning attention to this paper, which was published in 1973 in a comparative leukemia research. It was a summation, the final address to an international conference of cancer and leukemia experts given by Jay Clemenson. She was of the Danish Cancer Registry and Finnish Institute. She's a world-renowned epidemiologist. What she said was, on the next to the last page of this paper was, quote, "We are in fact establishing conditions for a possible pandemic of an oncogenic virus variant on the scale of influenza of 1918. So what she's saying here is that we're getting ready to cause a worldwide epidemic of a cancer-causing virus variant on the influenza pandemic of 1918. That pandemic of influenza 1918 killed millions and millions of people. She said, in tissue cultures, we have created conditions for propagation of viruses in cells from a host different from the original. In other words, we're taking viruses and growing them in tissue culture and changing the species that they infect. She said, quote, "This will tend to increase enormously the chance of mutation into variants acceptable to new hosts, such as man, and by their heterogenetic qualities, they may have neoplastic capacity in a new host. In other words, they may have cancer-causing capability in a new host." So she says, quote, "It is possible to visualize a mutation of a virus into a variety of high contagiosity to man, resulting in a pandemic, a worldwide epidemic of neoplastic disease, of cancer, before we could develop a vaccine." And that's really nothing more than what AIDS and the sudden explosion of so-called human retroviruses are. She says, quote, "What are the risks involved if some RNA virus or some DNA template would happen to infest some of the production of a vaccine?" So she actually tells us that what's happening would happen, and she tells us how it would happen by the contamination of a vaccine production by an RNA or a DNA virus. That was written in 1973.
And according to what you say, it is borne out.
Yeah, according to what we say, according to what, I think that people can determine that for themselves.
John, do you believe that this amounts to a prediction of AIDS?
Yes, I think that is a prediction that would cover AIDS as well as cancer and possibly a number of other things. Definitely was predicted.
How about you, Alan?
Not only was it predicted, but I said there were meetings that predicted that eventually one of these genetically altered viruses would get out into the environment.
It was predictable.
Absolutely,
without any question.
On a lot of levels.
You say that the AIDS virus was requested by whom and for what purposes?
Right. This comes from a bulletin of the World Health Organization, volume 47, 1972, page 257. And I'll read the third summary, but this was an article which was discussing the viral implications for human diseases. And under the recommendations, and the recommendation number 3, what they said there was the following, quote, "An attempt should be made to ascertain whether viruses can in fact exert selective effects on immune function by depressing 7S versus 19S antibody or by affecting T cell function as opposed to B cell function." What this says is that can we suppress IgG, which is an immunoglobulin, an antibody, or IgM, a different kind of immunoglobulin, or can we affect T cells as opposed to B cells? The possibility should be looked into that the immune response to the virus itself may be impaired if the infecting virus damages more or less selectively the cells responding to the virus. So can we make a virus that destroys the system that's supposed to protect us against that virus? We feel that this says, let's see if we can make AIDS. So is it a shock that we now have this virus running in humans.
And that's the World Health Organization.
That's written in a bulletin of the World Health Organization in 1972 and signed by many of their members of the World Health Organization.
Do you interpret this as a request for an AIDS virus?
No, I don't.
What do you think of it?
I think it's suggestive that they're talking about testing a virus or finding a virus to test to see its effect on the immune system, but I don't see that they're requesting what we say is AIDS, or that they're talking about widespread testing necessarily, or testing on humans. This could be in vitro testing in a lab. It's possible. It's not clear what they're saying there exactly.
Do you think it's dangerous to experiment to create a virus that could wipe out the human race?
Obviously.
Do you think it's irresponsible of the World Health Organization to even put out the idea?
Of testing a virus to see if it could wipe out the human race, yes, but I don't know that that statement is saying that.
Well, if you can wipe out the human immune system and create a disease that is contagious, against which therefore the defense to it is wiped out, then what are you doing?
You might be trying to test the effects of the immune system without creating a virus that would wipe out humankind. You might be trying to find out something about the immune system. From that statement, I don't see that it's clear that they're trying to create AIDS.
Well, they didn't have a name for it, but they succeeded in creating something that fit the description that they gave the name to.
I don't know that they did succeed in creating that. That's the question.
That's a question mark. Do you deal with that in your book?
Yes, I go into that somewhat in the book, that question of, is this created? Is biowarfare responsible? Dr. Strecker and I share a feeling about biowarfare establishments, I think, throughout the world, that they're very dangerous activities, very, very dangerous activities, and that lunatics mainly participate in those activities. Whether or not this specifically is talking about what we're talking about here tonight is another question I have my disagreements about.
What do you have
I
to
think
say?
you have to read between the lines. I don't think any journal is going to say, I think we should do human experimentation. I mean, no journal is going to print that. I don't think that it's a coincidence that this type of virus, which was immunosuppressive, had one of its origins in Africa, because we know that the World Health Organization uses Africans as experimental subjects when they go to test vaccines. That is a very common place to have pharmaceuticals tested, first in Africa, third world nations, and then later, if it works, in our country.
Right, I would suggest that people who really want to interpret this for themselves, read the article and make your own decision. I'm not trying to tell you what to think, but read it and then decide for yourself. And if AIDS fits the description, it fits.
And where are they going to get a hold of it?
It's in the medical libraries of the world, or you can request it from your library.
There's a good bibliography on that in my book, AIDS and the Doctors of Death, which basically take a lot of Dr. Strecker's references, quote the paragraphs that I think are significant, and then give the reference. It may be difficult to get it through a regular library. You most probably would have to get it through a medical library, and there we oftentimes run a communication gap because oftentimes the public is not allowed into medical libraries. But I'm sure with a little pressure you can get these references.
Dr. Strecker, during a workshop on histocompatibility antigens in 1972, officials of the World Health Organization recommended the study of the relationship between the immune system and the genetics of an individual by using vaccines.
Right, that's actually taken from a different paper. It's a pretty long paper. That was, here it is. And actually what this paper said, it's interesting because John was talking about whether or not there was human experimentation. I'll let you decide for yourself. This was taken from a conference sponsored by the John E. Fogerty International Center for Advanced Studies in Health Sciences and the World Health Organization. And at that conference and histocompatibility workshop, D.B. Amos of Duke University and others, alleged independent scientists, has suggested at page 1102 of this document the following. In relation to the immune response, a number of useful experimental approaches can be visualized. One would be the study of the relationship of HLA type, and H stands for human, not for bovine, cattle, sheep, monkey, anything else. It stands for human. Leukocyte antigen type, to the immune response, both humoral and cellular, and that means B and T cells, to well-defined bacterial and viral antigens during preventive vaccinations. That means let's test these things during alleged preventive vaccinations and we'll see how they're processed. This approach would be particularly informative when applied to SIB ships. That means let's do it in brothers and sisters of the same parents and see how this processing is different between the brothers and sisters. So you can say, well, that doesn't mean they're still going to experiment on humans, but HLA type stands for human leukocyte antigen. And in this document it says there was an intention of this study to be the time relationship between infection and antigen administration. Now, if you administer only part of a virus or a microbe, you never get an infection. So the fact that you wanted to study the time relationship between an antigen administration and infection implies to me that they were administering competent organisms or organisms that were capable of infecting people.
The World Health Organization recommended that viruses should be studied to see if they could destroy specific parts of the immune system. Why do you interpret this to mean that a virus should be created to do that?
Well, because at the time there were actually no virus. That's a very good question. Because at the time there was no virus that would do it. So if you look back, except in animals, and even there it wasn't truly competent or complete, there is no history of a virus that could destroy the immune system, particularly of man. So again, you must ask yourself, how does this new creature come into being? How is it that now it leaps forth? Where did it come from? How is this possible? And also, how could a virus, if you believe in evolution, how could a virus now exist that destroys the immune system that's supposedly been around for millions and millions of years, say? Why wouldn't the immune system have evolved to account for it? Or if you believe in creation, then why would God have made such a horrendous error and given us a virus that destroys the system that he designed to protect us? It doesn't make sense from either of those things. What makes sense is to say, well, this is something brand new that somehow came out of a creation that the immune system or hasn't been dealt with before.
You've done a really excellent job in presenting your thesis in the Strecker Memorandum, and I'd like us all to take a look at how you have presented at least a portion of the case that you're making now.
This is actually a tape that we have put together, the Strecker Memorandum, which is an hour-and-a-half summary of what we've been going over here today and before, which is available to everyone who's here in the audience or in the TV audience, either through Elizabeth People or through myself in Los Angeles, and they can contact you or me to get this tape directly. I think maybe we have a little clip here.
Yeah, we'd like to take a clip of it. This is the document that convinced me that you should be at Summit University Forum, and I think you'll all agree.
And the overall picture is that suddenly in humans we have an explosion of disease, not just AIDS, but other retroviruses, HTLV1, a T-cell leukemia virus, HTLV2, HTLV3, HTLV4, which is a new, recently recognized AIDS virus, also known as HIV2, HTLV5, which is the cause of mycosis fungoides, and HTLV1 lookalike, which looks like this. I call it 1 lookalike. Now, even looking at, say, let's throw AIDS out for a minute, you have to ask yourself, where did all these other viruses come from? And my explanation as to where these other things have come from, along with the AIDS virus, is the following. If you look at animals, particularly cattle and sheep, you'll discover an interesting phenomenon. In cattle, there's a virus known as bovine leukemia virus, which has the exact same morphology, which means shape, the exact same relative molecular weight, the same magnesium dependency, and the ability to produce B and T cell leukemia of cattle, and is proliferative in tissue culture. If you look at bovine syncytial virus, you'll find another virus of cattle, which has the same shape, the same magnesium dependency, the same basic appearance, and produces hairy cell leukemia in cattle. If you look back, you can discover a virus known as bovine Visna virus, which has the same appearance as AIDS, the same molecular weight, the same magnesium dependency, and in 1974, either 1 or 3 here, bovine leukemia or bovine Visna virus, was producing pneumocystis carinii pneumonia in chimpanzees. And if that isn't AIDS, I don't know what it is. If you look at the genes of the AIDS virus, the genes of the AIDS virus don't look like monkeys. The genes of the AIDS virus, in every paper published to date, look like the following: bovine leukemia virus of cattle or Visna virus of sheep. Now, these are retroviruses of animals, and these viruses are known to cause brain rot in sheep and to cause leukemia of cattle. So, is it possible, I mean, it was my question, is it possible to cross these two viruses and make AIDS? Now, of course, if you go down and ask your local AIDS expert, is that possible? Most of them will probably lie and tell you, no, that's not possible, that's just nonsense. But the truth of the matter is that AIDS viruses, in a sense, are much like humans, in that if you have bovine leukemia virus on one hand and Visna virus on the other hand, and you simultaneously infect a human tissue culture, what comes out of that infection is not only the original parents, bovine leukemia virus and Visna virus, but what comes out of that is every possible recombinant that will grow. In fact, not only that, also comes out of the ones that won't grow, which you can't identify because some of them down here are recombinants that come out, but they won't reproduce. They're called, in fact, in retrovirology, those are called incompetent. They're not capable of reproduction. You might call them impotent. They can't reproduce. So, we get out not only the competent, but the impotent. So, in fact, these viruses do, in a sense, not only reproduce themselves, but they make babies, which are different than the parental strains. It's just like each of us. Each of us is a recombination of our fathers and our mothers. If you say, well, we look at the AIDS virus, and we say, well, it's only 50% identical to Visna, and it's only 50% identical to bovine leukemia. And they say that's, quote, distally related. It's like saying I'm distally related to my mother or my father because I only contain 50% of their genes. Of course, that's nonsense. The fact is that containing 50% of the genes of the virus, you could be the direct descendant of the parents. It's the same as each of us. Each of us is a direct descendant of our parents, and we contain 50% of our mother's genes and 50% of our father's genes.
Thank you, Dr. Strecker. This film is so educational, so understandable, and you have such a talent for conveying the step-by-step understanding of the creation of viruses that someone with absolutely no background very clearly gets the message.
Right, I hope that's true. We spent a great deal of time and effort in putting this together, and actually my feeling is that anybody who views this can learn more about AIDS than probably 99% of the doctors who are walking around talking about it. So, as I said before, the listening audience can get it directly from you or from me, and we hope that they do, and we hope everybody goes home and shows it to everybody they know, and they show it to everybody they know and get this word out. And again, if you look at the bibliography on this tape, it's not New York Times. This is articles are directly taken from the medical literature, cancer research, virology, Journal of General Virology. I mean, and if you doubt that those articles are really there, then I suggest that the audience go to the libraries, pull them out, and read them and see what they say.
As you know, also available will be the full-length film of this forum, and so we have two ways of reaching people with this very important message. So I'd like to give you the opportunity now to go step by step through what you mean when you say that the AIDS virus was produced.
Right. Let me get back to the blackboard here. We believe that the AIDS virus, this virus, a D-type retrovirus, was actually produced in a laboratory. And this virus was produced in the following manner. As was illustrated on the tape, we believe that the virus came from a virus of cattle named bovine leukemia virus and Visna virus of sheep. If you look at the genetic structure of the AIDS virus, every paper written to date on AIDS says that the genes of the AIDS virus look like one of these two. So my simple question was, when I started to look at this issue, was, could you cross these viruses and make AIDS? And of course, as illustrated, most people say, no, that's not possible. That's the actual nature of this virus. These viruses recombine, they interact with each other, and produce not only new viruses, but recombinants or babies. So when you put these two viruses together in tissue culture, what comes out is not only bovine leukemia virus, the original virus, and Visna virus of sheep, but you get all the little babies, which are the children or the recombinants of these two viruses that will grow. And that is, in a sense, how we believe that this virus was produced. It could have been produced in a laboratory, where that combination could have occurred in a tissue culture, or it could have occurred directly in a cow, where if it was injected with sheep serum, that you could get out the recombination in the cow. And these viruses in cattle come out directly in milk of cows, in urine of cows, in blood of cows, in the meat, in the tears, in the saliva. So it's easy to see why cattle can infect each other. Now back to the question of how could it, if you produce this virus, how do you get it to jump species from running in a cow? This is a cow, bovine Visna virus. How do you get it to now run in humans? And the way that you get it to do that is by tissue culture in human tissue. And that was addressed in a paper that I have here. First one, this was entitled Common Genetic Alterations of RNA Tumor Viruses Grown in Human Tissue Cells. This was published in 1972 or 3 by 1971 by Stuart Aronson of the National Cancer Institute in Bethesda, Maryland. And what he showed was that growth of mouse retroviruses in human tissue would adapt that virus to grow in humans more, and such that it would now grow mostly in human tissue and no longer grow in mice. That experiment was expanded in 1973 in a very interesting, elegant piece of work by Maruyama and Domchowski from Texas, who wrote Cross-Species Transmission of Mammalian RNA Tumor Viruses. The very topic that we're talking about, how does a virus of one species get into another? Cross-species transmission of mammalian RNA tumor virus, and they showed that you could take a virus from, say, a rat or a cat or a mouse, grow it in tissue of another species, and then if you injected it into another species, you could produce disease with that virus, although it originally may not grow there. That was in 73. So that's how you adapt this virus of humans, of cattle, to grow in humans. In 1981, Cedric Mims, who was a member of the World Health Organization vaccine production program in the past, wrote an article in Microbiological Reviews in 1981 entitled Vertical Transmission of Viruses. And in that he said that a fairly common contaminant of fetal calf serum was an alleged bovine Visna virus. That was on page 278. And of course, fetal calf serum is the growth media, the growth hormone that they use in almost all tissue cultures worldwide. So what he's saying is that in 1981 is that we're putting into every tissue culture that we're growing in every laboratory throughout the world fetal calf serum, which is contaminated with a virus that was named bovine Visna virus. Now, the one mistake that they made, or perhaps one error that they made that's crucial to this discussion in the fetal calf serum, is that fetal calf serum is taken from fetuses of cattle and then injected into tissue cultures. So that the theory is that fetuses are sterile because there can be no organism contaminating the fetus. But in fact, these viruses, retroviruses, which are incorporated into the genetic makeup of various species, are transferred in the genes occasionally, and occasionally fetuses are contaminated by the genetic transfer of information from either the mother or the father. So those serums taken from fetuses are in fact not sterile. They are potentially contaminated, which leads you to the problem of contamination of all cultures grown in tissue. So,
And all studies.
right, of all studies and in viruses. Yeah.
So you're dealing with a factor that you're not acknowledging, and that's coloring every other experiment.
Right. That brings up another important little book. Let me just mention it here. This is a book called A Conspiracy of Cells
Excellent book.
by Michael Gold.
Right.
This is the story of Henrietta Lacks.
That's right.
Her picture is here in the front of the book, this lady. And so again you say, why is it that we have now AIDS 1978? Why didn't AIDS exist in, say, 58 or earlier? Well, in 1951, Henrietta Lacks walked into Johns Hopkins and died of a very malignant cancer. Her tissue was the first tissue that would grow in tissue culture that was human. Before 1951, they were unable to grow human tissue in tissue culture. So obviously any retrovirus that you were growing was not growing in human tissue. It was only growing in animals, which may explain the explosion of retroviruses in animals prior to the 50s or 60s. But now, Henrietta Lack tissue, which grew and was sent worldwide, eventually contaminated most of the cultures, the tissue cultures of the world. In 1981, a paper was written in Science which showed that 100 of 300 cell lines of the National Institute of Health were really contaminants of Henrietta Lack tissue.
That's right.
In other words, if you ordered from the NIH what you thought might be rat prostate tissue, it's possible that what they were sending you was really a highly malignant Henrietta Lack tissue culture line. So if you're an unknowing scientist and not too careful about what you do and trusted them, you might grow a virus in that thinking it was growing in rat tissue, and really it was growing in human tissue. You might accidentally be adapting it to grow in humans. So this is an excellent book, Conspiracy of Cells by Michael Gold, which delves into that question. And I can assure you that if they can't control their cells, they surely can't control viruses in their labs, which are billions of times smaller.
This book is so exciting that once I started reading it, I could not leave it. I could not put this book down. And what it is saying is that during a certain time period, all experiments in the cure of cancer were rendered useless.
Yes.
Because every single experiment was really this HELA virus from this one woman who had this very severe cancer, and what is so diabolical is how they figured out how to keep this virus alive for experimentation. Would you like to tell everyone what they fed the virus?
Yeah. Give the recipe. Alan has the recipe.
The recipe, you know, because you think of witches, you know, that are adding chicken bones and
ahead,
blood.
read the recipe.
The recipe from A Conspiracy of Cells is blood from human placentas. And the placenta is the sac that nurtures the developing fetus. And of course, it also contains powerful hormones and a host of viruses and bacteria, like Dr. Livingston talks about, as yet not fully investigated. That's the first part of the recipe. The second part is beef embryo extract, which is the ground up remains of a 3-week-old unborn cattle embryo, to which is added by going to a chicken coop with a big syringe, puncturing a chicken's heart, and getting fresh chicken plasma obtained from the blood of a live chicken heart. And that is the recipe that gave you the birth of modern day virology.
And that was determined by a lab technician assisting some people in Washington, D.C., Maryland area. And this technician would run to the hospital each time they had placenta and these things that were coming out of surgery, and grab them and run home and feed these viruses, cancer viruses, and kept them alive. And all of a sudden, all over the world, we're finding that this is such a hardy virus that it has wound up in most of the plates used for
It's cell line.
experimentation.
It's actually a cell line, not a virus, but it
The
grows.
cells, yeah, the cells, right.
Yeah, it grows throughout the world. It's contaminated millions of cultures, and the guy whose job it was to keep the cell lines of the United States straight, Walter Nelson Rees, who this book is about, was eventually defunded, and his entire operation closed out because he exposed the fact that billions and billions of dollars had been wasted in worthless projects.
In cancer research, this
Not
is
only
why.
that, but one of the experiments they discuss there is people testing normal human cells for their tolerance to radioactivity. And here were researchers in labs trying to figure out how much radioactivity human cells could stand. And they thought they were using normal human cells to test this, but they were using HeLa cells, Lax's cancer cells, right? And there was a speculation by at least one researcher that the figures that they would have come up with would have given too high a tolerance to normal human cells for cancer. In other words, they would have made a mistake and said, we can stand a lot of radioactivity, no problem. But they were really working with HeLa cells and they didn't know it.
And I think you were correct in your assumption too, which isn't even mentioned. I mean, God only knows how many viruses, were incorporated into that HeLa cell line, which they just assumed that was sterile. It's mind-boggling that experts could assume that you put all that junk in to grow those cells and assume that the cells are sterile.
Right, so you have a problem of contamination not only of the cell line, which may have been HeLa or some other amalgam, but then you're dumping in stuff that contains retroviruses and adding chicken blood and all. I mean, pretty soon you get to the conclusion. I mean, as I've always many times have said, you're surprised that this hasn't happened before.
Well, it sounds like arch fiends out of some laboratory of ancient Atlantis, reborn in Washington, D.C.
We're back, right?
So you are affirming that this was one of the breakthroughs whereby viruses, animal viruses, are now cultured in human tissue.
Well, they've been cultured in human tissue, even though they deny it. I mean, there are published AIDS experts who say you can't grow Visna in human cells. They grew Visna in human tissue as 20, 30 years ago. So they've been growing them there for 20 or 30 years. And the question here, too, is if you allow scientists to make these new organisms, let's look at the question of AIDS alone. Let's just suppose for an instant that AIDS came from nature just as they say it has when we believe it hasn't. Has any investigator of the AIDS virus been accidentally infected with it? Absolutely. There are at least 16, perhaps hundreds or thousands, who knows. But we know of at least 16 documented people, medical investigators or AIDS researchers, who have been accidentally infected. So if you tell me that you're going to make a virus in a laboratory that's brand new and nobody's going to get it, then that's obviously not correct. You can't control these things in your laboratory, because there are accidents that occur. People break things, they cut their skin, they get injected, infected. I mean, AIDS alone, the fact that medical researchers have been infected accidentally, proves that control of new agents in a laboratory is next to impossible, if not virtually impossible.
Well, they made the point that the HeLa cells were so hardy that they actually would contaminate by being airborne.
Right, yeah. Well, that's another thing. I mean, they say that this is not possible, and yet the HeLa cell compared to an AIDS virus, it's millions of times bigger. And so when they were opening their tissue cultures, they were aerosolizing HeLa cells into the air. They would float over and drop into another culture and take over and amalgamate with that culture and then become an amalgam or a recombinant between the new one and the HeLa. And they were very hardy and very malignant, I mean highly malignant. So obviously any retrovirus that HeLa was containing within her genes would be carried along right with her, just as she flopped over or happened to float in otherwise. So the combinations become pretty horrendous.
Well, the joke on all the scientists was that the Soviets presented us with some very special cancer cells, and upon examination, they were the HeLa cells, had made their way all the way from the United States through Europe to Soviet laboratories, given back to us as their original experiment.
Yeah, it's a very interesting gift. They gave us 6 human cancer-causing viruses, which turned out to be nothing more than monkey viruses growing in HeLa tissue.
Well, so much for the mad scientists of our time. Do you want to add anything more to the story of the production of this virus now?
Only the fact that, yes, something else. Let me just quickly say that this was actually 1974, which was ultrastructure studies of a Visna-like syncytia-producing virus of cattle with lymphocytosis. Well, really, what this paper in 1974 draws hand-drawing pictures of the AIDS virus and of the bovine Visna virus. And the interesting thing is that virus is the exact same morphology. In other words, it looks identical to AIDS. It had the same molecular weight. All the characteristics are the same. And then in 1978, they grew that virus and published in another paper in human tissue, human malignant bone marrow tissue. And so you can see how perhaps bovine Visna virus may have been adapted to grow in humans by growth in malignant bone marrow tissue. In 1974, in a paper published by Cancer Research by Harold McClure and a group of others in Farrar, this was a paper which was entitled Erythroleukemia in 2 infant chimpanzees fed milk from cows naturally infected with bovine C-type viruses. A complicated title, but the actual summary of this paper says that when they fed cattle viruses to chimpanzees in 1974, those chimpanzees died of a new leukemia and pneumocystis carinii pneumonia. And of course, pneumocystis carinii pneumonia is the hallmark of AIDS. It represented immune destruction. So it's our feeling that that was already illustrating that this virus could have produced immune destruction, perhaps in chimpanzees, as far back as '74.
As we talk on about this, discussing the technical angles and the science of it, we have to stop for a moment and realize that we're talking about something that has already been unleashed, that can no longer be stopped, and that is likely to spread throughout the planet.
That's correct. I personally feel there was very unlikely to be an allopathic cure for this problem. I think, maybe we can get to that near the end, but we can get into what I think this might be cured by. And I believe that there is a cure perhaps available if it's developed, and may have been available as far back as 1925. And it was created by a guy named Raymond Roy Rife, who was a garage tinkerer and inventor, who invented a way perhaps that these viruses could be cured.
All right, we'll get to that. The London Times, May 11th, 1987, ran a front-page story which said the AIDS epidemic may have been triggered by the mass vaccination campaign which eradicated smallpox. I'd like you to comment on this article and explain why you think the virus was deployed.
Well, 2 reasons. One, it could have been accidental or intentional. If it's intentional, it's clear-cut. Accidental, or it could have occurred by contamination of vaccine production, or as in a sense, the vaccine, smallpox vaccine is prepared directly from cattle. Cattle's bellies are shaved, sliced with a razor, the vaccinia is dripped on. They put a cow in stanchion so they can't lick. A few weeks later, they come back, put a stainless steel container underneath, scrape off the scabs, collect the effluent, dry it out, and that's your next smallpox vaccine. Now this virus, which can obviously grow in cattle, bovine Visna virus, could have been a contaminant of cattle, it could have been a contaminant of the smallpox vaccine, it could have been inoculated accidentally into millions of Africans, just like simian virus 40 was inoculated into a million Americans in the polio vaccine in the early 1960s. John actually researched that publication. It was Monday, May 11th, 1987, and very few papers in the United States published anything on that.
Why was it ignored in the U.S.?
Well, I think that indicates that anything that does not go along with the government view, you know, a simple little monkey virus, and anything that challenges that, like maybe it didn't come from the monkey virus, maybe it was connected with vaccines, is like suppressed across the board.
I've tracked down several articles on AIDS that have appeared in other countries that have been blacked out of the American press. And I wrote about the fact that that article in the London Times was blacked out of the American press. And I called AP in New York to find out why they hadn't received it from London or distributed it throughout America, because obviously they would have gotten it. It took me quite a while to find out, but the final word was from the managing editor of AP in New York. He said, well, why do you think this is a big story? I said, well, let's see. Front page London Times, they're saying smallpox vaccinations are going to kill millions of people, and you ask me why I think it's a big story. Right? Now, I'm not saying I agree that this is true, but we're just talking story here, as they say in Hawaii, talking story. He says, why do you think this is a big story? And I said, well, you know, so that's what I said. And then he said, well, let me put you on to the science editor. He could tell you what happened. So I talked to the AP science editor. I said, did you get a dispatch from London, from the London Times, through your AP office that talked about this story? He said, yes. I said, did you send it out from New York? He said, only to Boston. I said, why only to Boston? He said, well, I actually haven't read the original article in the London Times. That was his only answer. I said, well, why don't you read it? He said, all right. And of course, he never read it, or if he did, they never sent it out. The story never got anywhere. It never moved in the United States. And if you know anything about the news business, if you can imagine a front page article in the London Times saying something as dramatic as this, not appearing in an American newspaper, this is like, you know, something came from the moon, a UFO came down, landed on the front door, and they said, well, let's not write about it, you know. It's about on the same order as that. So obviously they had to say, no, we don't want to write about that, you know. We're not going to put that in the paper. And I talked to a number of people from newspapers about several AIDS articles, very dramatic, anti-establishment views. None of these views actually, as I write in my book AIDS Inc., do I agree with, but they are substantial views that were published in other major newspapers. They never made it into the United States.
So what's the symbiotic relationship between the science press and the establishment of medicine and the federal government and the moneyed interests involved? I mean, why doesn't the dignity of the press maintain itself as representing one quadrant of our sacred freedoms and being the vital voice on behalf of the people? I mean, who's paying these guys off? Are they just brainwashed?
Their paycheck is paying them off. That plus the fact that they, and I have a whole section in the book here called AIDS in the Media, and the way the media works and how they handle this. And I think it's very simple. I think that maybe there was a time once in this country when if you owned a newspaper and you had a certain point of view, you had to kind of make sure that your writers followed your editorial policy. Well, our society has evolved to the point where nobody has to say anything to anybody. Newspaper writers that write for the LA Times know exactly what they can write about and what they can't write about. They know if they cross that line, the paycheck is in danger, and they may get fired or they may get reassigned. They don't touch those stories. And most of all, they don't touch a story that requires any independent research on their part when it comes to science. This is verboten if you write for a major daily newspaper in America. No science reporter can do his own research. He merely reports and collates information from the press people at the federal health agencies who pass this information down the line. That is the function of those people, and they understand it very well. Plus, you have major pharmaceutical advertisers in these newspapers, and so if the New York Times decides to fool around with the idea that vaccines, you know, are giving people lots of health problems, serious, fatal health problems, and they happen to have a pharmaceutical advertiser that manufactures vaccines, they may lose a lot of money. Pretty simple.
So you exercise your freedom of the press and you write your own book.
You got it.
Can we see it?
And I'll add something to that, and that is, in order to get this book written, it's published by the Human Energy Press, and it's available from them. They're in San Bruno, California, or it'll be in local bookstores around the country, probably in the fall, at least, if not before. I got this publisher who was interested, and I said, one condition. I'm the editor and the writer. Because I knew that if I were assigned by a major publisher to do a book like this, It just doesn't work. You can't get it by these people. They will chop you down to the point where what you're saying is just, packaged oatmeal. If you're the editor, though, as long as you're not, libelous, you can say whatever you want. So that's the only way this book would get published.
Thank you for having the courage to publish it.
Let me say one thing. I've sort of worked with John on and off. He's reviewed a lot of our information, and the interesting thing about it is that I'd give him a reference. He would come and, get the actual reference itself, read it for himself, and make his own conclusion. He obviously doesn't agree with us on a great many things, but he read the material himself and he made his own decision. Most so-called reporters simply call up an expert and say, what does this mean, or what is your opinion? And they report to the American public that expert's opinion. They don't take the time to go down and read anything for themselves. I wish that they would, because we wouldn't have some of the ridiculous stuff that we have that's coming out. But it's not entirely a fault of the reporters. Some of the problem, in my opinion, is still left with the American public, who allow this to happen and who have, in a sense, stopped reading for themselves also, and don't rigorously question everything that they read in a newspaper. I mean,
I also have great respect for John, and when I wrote my new book, which came out this year, I thought of all the people that I would really like to have to write an introduction to the book, and I chose John, so he's also in my book, AIDS and the Doctors of Death.
Well, yours was another book that I couldn't put down, and I don't read that many books cover to cover.
I don't know, you read them all.
Well, once I got into this, I really got into it. Now, what is your opinion, Alan? Could the spread of AIDS virus have been accidental?
Well, I think if it was an accident, that the accident was tremendously covered up. And it's like, if you see a man driving down the street drunk and he's hitting somebody, and there are 10 people there with a pen and pencil who take the license plate down, and nobody reports it, and the man hits the person, drives away. I mean, I don't think that's accidental. So I think that if it was an accident, somebody would have said, maybe it's an accident. But the fact that no one has ever said that, on the basis of all this evidence for biological warfare and all the horrendous things we have already done on the planet to our brothers with biological agents.
Well, what do
I don't think that it isn't. I don't think it was an accident. I think it was deliberate.
Like, how could it be an accident when it is such a select group of people in select cities?
Exactly.
A select age group.
I mean, how come it affects, black Africa, with all the problems they have there and all the racist stuff going on there? So, I mean, and then we know that they experiment there. I mean, that's the one place on the planet where scientists experiment with human beings the most. And how come it started exclusively in the most hated, minority in the world, in the country, and also probably in the world? But if you're diabolic and you want to start a biological warfare experiment, I couldn't imagine a better group to start with than the gay population, because you inject them, you sit back, you watch it spread, and it's just mind-boggling, because you know that people are going to blame their sexuality and their lifestyle. So it's like a perfect cover-up. So, I mean, I think it was a horrendous act, and I think the more you think about that, the more you realize that that was no accident of nature. There's no way that that virus could have gone to Manhattan in that group.
Manhattan, San Francisco,
I'm talking about
Los Angeles,
primarily, I mean, initially it was Manhattan. I mean, it was like, the same Manhattan borough that the hepatitis blood experiment program started in 3 months previously. I mean, is that a coincidence, or is that a coincidence?
And
So I don't think it was an
name the
accident.
group that was selected, the character.
Pardon?
Name the group, the the characteristics of those.
The group that initially came down with the disease were
that
white.
that were given the hepatitis B.
Yeah, they were white. They were well educated because they wanted to follow the people and make sure they got all the blood specimens. They were well educated, they were healthy, and they all lived in Manhattan. And they only wanted people under the age of 40. They didn't want any old people to mess up the experiment. And they didn't want you if you were not promiscuous.
They wanted you bisexual, homosexual?
Yeah, they didn't want any heterosexuals. And they didn't want anyone who was monogamous. Because the idea was to test this vaccine. And I don't think it's a coincidence that then a few months later, a bunch of people in 79 and 80 and 81 developed this disease who were white. They were from Manhattan. They were previously healthy. They were well educated.
Well, they were part of those who were vaccinated, weren't they?
Well, somebody would say, well, no. But the profile of the people that signed up for the government test and the profile that ended up with the disease initially are the same. It's unbelievable.
Let me help Alan out. Maybe it'll be better if we draw a picture. Sometimes a picture helps. Here's what he's trying to say. In 1978 in New York City, young white male homosexuals were recruited who were promiscuous, who were between the ages of 20 and 40, to participate in the hepatitis B vaccine study. That was in 1978. In 1980, the same group of people were recruited in Los Angeles, San Francisco, and then other groups were in Chicago and St. Louis. Now, retrospectively, where did AIDS break out? AIDS broke out where? New York City, Los Angeles, San Francisco. 78 in New York City, and young white male homosexuals who were between the age of 20 and 40 and were promiscuous. 1980 in San Francisco and Los Angeles, and young white male homosexuals who were between the ages of 20 and 40 who were promiscuous. Now, if you look back into San Francisco, interesting data. 6 of the first 10 AIDS cases in San Francisco came directly from the hepatitis B vaccine project. The hepatitis B vaccine project was conducted on homosexuals to prove that the hepatitis B vaccine would prevent the development of hepatitis B. 6 of the first 10 of the first 10 AIDS cases in San Francisco came directly out of that project. It's in the medical literature and anybody can look it up. In 1986, published in the Journal of American Medical Association by Claude Stevens, who took over this study for Wolf Smootness, who conducted this study. And Smootness was from Poland, was educated in Russia, came to the United States as a immigrant, and rose rapidly among the ranks of virologists and became the director of the New York City Blood Bank. It was he who set the criteria. Now, in 1986, Claude Stevens published this graph which showed that in 1984, approximately, was this 84 or 83, Alan?
84.
84, approximately 50% of the people in the hepatitis B study had positive for AIDS, or HIV positive. 1984, and it was a straight line increasing. Now, it's now 1988, which would be out here if it continued up at the present rate, provided for some miraculous reason it didn't level off. You could predict that 100% of those people in her study should be positive for AIDS. The interesting part was, the part that's most interesting, and which no one has discussed, is that this is not cross at zero. She didn't say that none of them had it. In 1978, she says that 6% of the group of her study were positive for AIDS. Now, of course, that proves that her study could not be responsible for the pandemic if they was already there at the time the study was initiated. She says that was taken on blood drawn in 78 or 79. And she doesn't state whether it was 78 or 79, but let's just say for the moment that it included 78. Obviously it was 79, it doesn't prove anything because you don't know. But 6% of 1,000 was approximately 60 people. Now if you back calculate to how many people in New York City had AIDS in 1978, it couldn't be more than 100 at most. So, if 60 is in this study, my question to the medical establishment is, What is the third factor that's responsible for this, besides the fact that they're HIV positive, besides the fact that they're in the hepatitis B study? There's obviously some factor that's giving them AIDS, and what is it? So, I conclude that there's no doubt there has to be some kind of relationship, in my mind, between the hepatitis B vaccine study and the outbreak of AIDS.
And the government has never investigated that. I mean, wouldn't you think the first thing you would do, or one of the things you would do is investigate. I mean, they have all this blood. They kept calling them up every 6 months to get more. Wouldn't you think when the AIDS epidemic broke out, they would keep close tabs on those men to find out what happened?
I can fill in a little bit here. Before the virus HIV was announced as a cause of AIDS in 1984, there were a number of doctors that were warning people in writing that maybe there was something the matter with the hepatitis B vaccine, that maybe it had something to do with AIDS. When Robert Gallo announced that HIV virus was the cause of AIDS in 1984, then certain researchers said, oh, well, now no problem, we can really find out if there was a connection by simply looking at the vaccine and seeing if there was HIV in the vaccine. We can track the people in the study, we can do all of these things. They then went on to write several papers exonerating the vaccine, saying we know it had nothing to do with human illness or AIDS. When you read those papers, and I won't try to go into detail about them, but there's a whole appendix in the book about that attempt to whitewash the vaccine and say it was fine, you see glaring problems right in front of your face immediately. The first of which is they don't test every available amount of the vaccine they can get their hands on. They spot check it, which is so bizarre. I mean, it's as if to say, well, let's look and see if there's, are there any germs on the table here? Well, let's look here and here. No problem, there are no germs on the table. Well, why don't you look at the whole table? And these vaccines are made in pools, big, large amounts of blood and then vaccine and so forth. They did not really do a thoroughgoing analysis from all of the literature that I can find of as much samples of vaccines as they could get their hands on. That's the first thing. The second thing was, in the graph that Bob just drew, Clad Stevens, who works as a researcher at the New York Blood Center, she did a follow-up study attempting to exonerate the vaccine. And she said, look, the people who got the hepatitis B vaccine later went on to test positive for the HIV virus at a certain rate. The same rate that people in that hepatitis B study did who took the placebo. In other words, who didn't even take the vaccine, who was just injected with water or whatever. Therefore, we know that the vaccine was no more harmful than the placebo, because these people who were taking the placebo over years later began to also test positive for the virus at the same rate. Therefore, no problem with the vaccine. In the next paragraph, she says, of course, the group who took the placebo and the group who took the hepatitis B vaccine are not really comparable in a sense, because we know that the group who took the placebo had more sexual encounters than the group that was taking the vaccine. So all of a sudden she's now saying the two groups are not equivalent, therefore the whole study is skewed, therefore the whole attempt to say that there's no problem with the vaccine just falls out the bottom. And you would think, God, any researcher should be able to figure out they just, blew their own game here, what's going on? No explanation, no nothing, and the matter has been more or less dropped. And like so many other things on this AIDS business, researchers just say, oh, well, I read this paper in the journal, it's proven, no problem, the vaccine is okay, everybody can take it.
Right. Another interesting thing in this study too was that if you read the side effects of the hepatitis B vaccine, you see what we know today from retrospective analysis is that supposedly a bunch of these guys were becoming AIDS positive from 1978 on. And what is the acute AIDS infection characterized as? Big swollen lymph nodes, fever, sick. Well, How come none of those people had any of those symptoms during the hepatitis B vaccine study? Did all these doctors that are going over these patients meticulously, just manage to miss the fact that these guys have big nose, they have fevers, and they're sick with an acute mononucleosis type syndrome, which is what AIDS is acutely, and yet if you leave the side effects that developed during the hepatitis B study, none of those findings appear in the explanation of side effects or the reactions that occurred as a result of the vaccine. You see, because they would have been observing it as a reaction of the vaccine, not as a other contamination. So you wonder how that got sort of swept under the carpet too.
And then also the group that got the placebo, they told them, as soon as the experiment is over, we'll then give you your injection. So, I mean, you're going to wreck the placebo group anyway.
That's right.
Because a year later, they injected the placebo group. The guys
there should have been a bunch of symptoms in the placebo group, which they knew had nothing. So you wonder, why was there not any explanation of a group of symptoms that appeared in the placebo group? It's very peculiar.
The bottom line is that it just was really bad science, and any researcher, I mean, I'm a reporter, I'm not a scientist. I'm fond of telling people who say, what are your credentials? I say, I have no credentials. Absolutely none, except that I can think. And the reason that I'm criticizing certain aspects of this whole thing mightily is because the thinking is so bad that anybody can notice it. And when you read these papers, you know the thinking is very, very flawed. With an elementary course in logic, anybody can look at that work and say, there's a contradiction here. This doesn't follow from that, but yet within the scientific establishment there is such pressure these days to agree, to go along with the crowd, to get research money, to be on the right side of all of this, that scientists just, you know, they salute and that's it.
And the physician that ran the trials in the West Coast was the same physician who was doing animal experimentation a few years previous to the hepatitis B vaccine program, where he was deliberately infecting animals by getting cats and so forth together, and then they would pass on their cat, retroviruses. And he was saying all the while that he was doing these cat experiments, someday, soon, you're going to realize that these germs that we transfer back and forth in our lab between these animals, they cause cancer. And one of these days soon, you're going to see the same thing in human beings. So a few years later, he's the guy that they chose to run the experiment. I mean, is that a coincidence? And then you wonder why all these animal people, have risen to the top, of AIDS research. And here I am, I think I know something about AIDS because I've been studying the cancer, right? But I mean, they're not calling me. They're calling some animal doctor. And I'm going, how does an animal doctor know all this stuff about human beings that I don't know? I mean, it's so bizarre.
Indeed.
Does anybody here believe in evil?
I do.
There's a lot of it around.
These are not unintelligent people. You say the thinking is flawed? Come on.
Oh yes. No, I think that there's some of them where, I believe that the Nazis had a certain system. That system is probably as old as the world. It's carried on all over the place, and part of that system is the bureaucracy. If you don't have a bureaucracy, the evil people don't have the machine to carry out what they want to do. In the bureaucracy, part of the credential that you get to say, I am a member of the bureaucracy is, whenever you are confronted with evil or something horrible, you pretend you didn't see it. And there are many of those people running around all over the place in the scientific establishment today, particularly in the biowarfare community. Their job is to not see things. They are not the shakers and the movers. They are they're the machinery that carries out intentions of other people. So yes, there's definitely evil, but there's also, as I think it was Hannah Arendt said, the banality of evil. The banality of evil is the person who goes along with the orders, who convinces himself not to think. So
the agent.
The agent, exactly.
The facilitator.
Facilitator.
The passive person.
Sure.
That you want to punch in the nose.
That's right. often the most arrogant. When I've talked to lots of these people on the telephone. I've spent several $1,000s talking to people doing research and science around the country. And if you didn't know anything about what you were researching, I mean, if you didn't even know disease from AIDS, from monkeys, from mice, from anything, if you just listened to the sound of these people's voices, that's all you'd have to do. One of my policies as a reporter is sometimes don't pay attention to the words, listen to the music, that's all. If you just listen to the music of these voices, you know you're dealing with some very weird fellows. Very weird. They believe that they know. If you watch these people, I call it, I know I'm sitting here with two doctors, but I call it the doctor knows best syndrome. You know? And these guys are not these kind of doctors, but there are doctors, and when you get to be a medical researcher, you've graduated, you see, way beyond the doctor knows best to the second power. You're now into the 100th power. This is the attitude that says, you couldn't possibly know what I'm talking about. So I'm not going to waste my time on you with any specifics. Just take my word for it that you're either wrong or crazy. And most people say, yes, doctor.
Well, you're talking about a war, and you're talking about deliberate actions. And if we deal with AIDS, biological warfare, chemical warfare, on these terms, then we can attack something and we can move against it. But if we continue to believe that these are misguided individuals, they don't know what they're doing, they're making honest mistakes, and we'll try to tell them the truth, we've missed the point of the conspiracy that is really an international one, which I call the international capitalist communist conspiracy, because it seems to be evil minds on both sides of the fence, playing the people of the planet against one another for the changes they work to effect control through war, through manipulation of money, and so forth. But manipulation of population has been their game for thousands of years.
Well, if manipulation of a population is the game, I mean, you couldn't pick a better agent.
This is it.
This is it. It's the perfect agent.
This is population control.
Right. It gives you the power to manipulate everything, practically. I mean, and you see, one of the true dangers of this virus is, in fact, if nothing is done or there is no fix that comes to fixing it, then if the disease process worsens, as I believe that it inevitably will, then sooner or later the people sort of cry out and say, you know, do anything that it takes to save us. And then when that happens, that's when you give up all of your freedoms that we have won that are so dear to us. And that is, in fact, the most dangerous thing, in my opinion, of this epidemic. I mean, so that is, you know, if the manipulation of people is the game, then we have to be very suspicious about this virus on that ground. And, you know, it gives them awesome power if we, and particularly if nothing gets done to stop its spread, or is it spread and people don't know the true origin.
We have interviewed at Summit University Forum a defector from Eastern Europe, highest ranking military defector by the name of Jan Shena. And he speaks of being present when Khrushchev announced chemical warfare against the West, specifically the United States and our armed forces, through the purveying of drugs. And so the international drug games began, and suddenly, overnight, there were pushers outside of every army base, military installation throughout Asia. And he documents this very carefully. He was there. And what we're saying here is a decision was made by the Soviets to wage chemical warfare against the West. Now, this can be considered chemical or biological warfare. That's why I'd like to talk about Wolf Smunis coming out of Eastern Europe out of the Soviet camp and somewhere into a military establishment involved in just these things we're talking about. And I want to be sure you've had your final word on Wolf Smooness.
Smooness, right. Well, Alan actually knows his history better than I. Perhaps he could sort of outline it, and then I'll fill in any gaps if there are.
When he was a young man, he was a medical student. And during World War II, when the Nazis invaded Poland and split it down the middle, unfortunately he was Jewish, and his parents, his family were all killed in the Holocaust. And this is the story that appeared in a sort of an obituary type article after his death. And apparently he then was taken by the Russians and sent to Siberia, where after the war he was let out and allowed to go to medical school in Russia, in Tomsk, during which time he apparently married a Russian woman and had a child. And then eventually he was allowed to go back to Poland, where he worked as an epidemiologist, apparently very hard with an interest in hepatitis. And at one time he requested a leave, at which time the communists sent him to this particular recreation center where his roommate turned out to be the future Pope, the current Pope, John Paul. And after a while, he was getting a reputation in hepatitis, and he was sent to Italy to a big medical meeting, apparently accompanied by his wife and child, at which point he defected to the United States. Now, I'd always heard that it was difficult to defect, and especially difficult to defect with your wife and child, and that they oftentimes will separate families in Russia or in the communist countries to prevent such defection. At any rate, he arrived in New York City penniless, was able through some contacts, though, however, to get a job at the New York City Blood Center, at which time he worked his way up very, very rapidly to having his own laboratory, given millions of dollars by the government, becoming a professor of epidemiology at Columbia University. Then he was given charge of this hepatitis B vaccine and other vaccine programs involving Africa. And at one point, he was so high on the scientific genius scale that the Russians invited him back to Moscow to give a paper. Well, of course, apparently he was scared to death to go back, having defected. Why would you want to go back? Nevertheless, through his government contacts, he was able to go back, and apparently he was well received and came back. And then in 1982, he died of cancer. I don't know the type. And I always thought it was very strange that somebody with that kind of story, would be chosen to lead such an experiment, which after his death was glorified as one of the most important experiments in the entire world with global implications. And I think only I understood why that was. And I couldn't understand why, out of all the epidemiologists, and God knows they're all over the place now, why would they choose somebody like that?
So he is the very one who
Well,
conducted
he is the
this
one, and
experiment.
there's others that, that after...
In New York, this whole thing.
Yeah. I thought that was such a strange story, and I've been criticized as, being irresponsible for repeating that story, but I just think that's sort of a James Bond story, and I haven't read many stories like that in medicine.
Well, if you were he, how would you have managed to surreptitiously insert this virus?
Well, it's very simple. You have a bunch
vaccine?
of people, and you have the injectable, and you have the placebo. And all you have to do is, when you're giving the vaccine, is to have somebody come along and put something in everything. Because even though you have a placebo, who says you have to use it as such? If I were going to do a biological warfare experiment, I would put it in the placebo and in the experiment. That way you could say, well, both of them were the same. Therefore, everything is, we had a placebo group and a regular group, and there was no difference. But if you put the agent in both, it would always turn out the same. So, it would have been very easy for someone to come along and put an AIDS virus in there. And then after all, you're a doctor, you work in a clinic, and this comes from the government, and they say, this is the stuff to be injected. You say, thank you, you inject it, goodbye, and I'll see you in 6 months. Also, so, it's very, if you want to be diabolic in medicine, if you come to my office and I'm a mad scientist, I can put all kinds of germs in what I'm going to give you for your health. You come to a doctor not expecting that I'm going to hurt you. And I would hope that there are no doctors like that. But I know, I mean, you know from all these books, you know, that there are doctors that will take people, especially people that they don't like, and who feel that are an abomination, or, you know, that God hates them or whatever. And they feel that they're lower than animals and that they're expendable, and plus, but at the same time they serve a purpose, because, let's face it, the AIDS virus has been the greatest boon to cancer research ever. Because through AIDS, the scientists who've always wanted to prove that these viruses caused cancer in humans through AIDS have proven that. And all the people that were involved in the animal experiments have come to the top.
That's what they were all trying to prove.
Now, is that a coincidence?
That's right.
To me, it's once you open your eyes, you say, God, now I understand what's going on. It's all very simple. It's like when I first met Strecker, and I'm saying, well, how did they put it into the gay community? Or how did the gay community get the disease? And he said, it's very simple. They put it in. And I go, oh. You know, they put it in. You know, it's like it's so simple that you go, And I think that they did. I mean, I honestly do. And I think that you have the circumstantial evidence and the environment, you know, to show that theory, I mean, it's still a theory, it certainly makes more sense than the green monkey, which has now been thrown out. So what's going to replace it? I personally think this is going to replace the green monkey theory.
Do you wish to add to this topic?
Actually, yeah, very simply. I mean, you don't, if you have, first off, the way they recruited the people, it's not surprising that, you know, if some guy's at the New York City Blood Bank and who's promiscuous, he might have had a few of his friends show up. So if you look at it from a biological warfare viewpoint, you don't have to put it in the whole vaccine. You only have to put it into maybe one vial, or even a small portion of one vial into both the placebo and the control group, or the vaccine group and the placebo group, because say 3 or 4 people get it. Since it's infectious, it's going to spread. And we already know they picked them for promiscuity. So if you're monogamous, you're not in this group. So if you get an infectious agent, it's not surprising that your friends might come down with the infection. So even analyzing 99 out of 100 bottles and they're all free doesn't prove that the agent was not there. More importantly, these viruses, besides making recombinants of this nature, actually rearranging their genetic information, do another very interesting thing. They do genomic masking, or what's called pseudovirus formation. And this is particularly dangerous for the following reason. What retroviruses can do, in the combination with other viruses such as measles or vesicular stomatitis virus, when you co-infect a cell with, say, bovine leukemia virus of cattle and vesicular stomatitis virus, which will infect practically any cell in the world, you get out the recombinants that have vesicular stomatitis virus capsule on the outside and bovine leukemia virus genes on the inside, sometimes called wolves in sheep's clothing. And it is the capsule that determines what this virus will now infect. So even though the original bovine leukemia virus may not infect a particular cell type, the pseudovirus formed by the combination of the bad gene on the inside and the quote good gene on the the good gene part on the outside will infect any cell practically known. And so if you were designing a biowarfare project, you would of course put your bad news on the inside of a very benign coat and put it into and contaminate your culture or your vaccine. And of course, the only way then you could ever find out it was there is by growing it out. Because if you just tested for it, you wouldn't find it because it's covered up by, say, hepatitis B or something else.
That's the answer I was looking for. Thank you. That is just amazing.
They're doing it all the time.
Based on what you know, you are stating that mankind is threatened by the HIV virus.
Well, mankind is threatened by not only the HIV virus, but the other so-called human retroviruses which are out and running. HTLV1, T-cell leukemia virus, which has infected a large population of southern Japan, a large population of Okinawa, parts of the east. HTLV2, hairy cell leukemia, HTLV4, which is now all over Africa and beginning to be in Europe. HTLV5, mycosis fungoides infection, and a whole host of others. So even by itself, AIDS represents a threat to the existence of mankind, and that's without all these others, which may be, in fact, more infectious.
For which there are no cures.
There are no cures.
Why aren't we hearing all of these spoken about equally? Why is AIDS the only thing flashed in all the media?
I personally believe that because the speed of the AIDS virus exceeds the speed of these others, so that it takes much longer delay between the time of infection with, let's talk about HTLV1, human T-cell leukemia virus. HTLV1, you get infected today, the average lifespan before development of disease is about 15 years. So 15 years later you get the disease, and on the average, say 6 weeks later, you're dead. So because AIDS acts so much quicker, even though lots of people may be infected with one, we haven't yet seen the explosion of the disease. That's because we're only seeing AIDS because it acts, say, in 5 or 7 years. In another few years, I think, and I predict, that you're going to see an explosion of T-cell leukemia among humans. If you look at AIDS people, about 20 to 30% of those who carry HTLV3, the AIDS virus, the putative virus, also carry as a contaminant, or with it, they carry HTLV1, or T-cell leukemia virus. So how is it that these people who are carrying AIDS happen to be carrying this other human retrovirus? Of course, they die of the AIDS infection before they ever get a chance to develop the T-cell leukemia. The same thing in a sense is true. It's like if you look at Visna virus in sheep. On the western range of the United States, approximately 75% of the sheep have the mighty Visna in them. But the fact is we end up killing them and eating them before they die of Visna. So we keep producing more and more sheep that have Visna in them, or at least the same percentage, but all of them carry it. If you left them out there long enough, they'd all die. The same thing like chickens. We mass produce chickens on a giant scale, all of whom are infected with this leukemia virus that we're trying to eliminate here. But we eat them before they get a chance to develop the leukemia.
And what happens to us?
We're potentially exposed to these agents. So in the case of, that isn't to say that everybody who eats chicken is going to get a leukemia. That's not what I'm saying. But there is in fact, there is some connection between these agents and the development of cancers and leukemias in humans. With HTLV1, I believe that a very high percentage of those people who get that virus will subsequently develop T-cell leukemia.
So people who are eating lamb or who are eating chicken, as this goes through generations, does there come a time when that virus is playing itself out in a fatal disease in them?
We're going to find out. I don't know the answer. I mean, that's a difficult question to answer at this point. The one thing that protects us against that is the species barrier. So if you're exposed to another species virus, which hasn't had a chance to adapt itself to humans so far, you don't seem to get that. Now, this brings us back to the question of pleomorphic organisms as the true cause of cancer versus viruses as the cause of cancer. And as I've said before, that's where my thinking kind of grays out, and where I'm not entirely sure of what that relation, I mean, I don't have decided yet what that relationship is between the pleomorphic organisms, cancer, these viruses, and humans. But let me tell you, this I know for sure. HTLV1, 2, 3, 4, 5 are all infectious human leukemia agents or AIDS agents, and if you get them, you run a big risk of getting either AIDS or leukemia.
What I'd like to tell you and our audience about our chickens and turkeys at the ranch is that one of the main reasons that we have our own chickens and turkeys is that we do have people who enjoy that meat. And it so happened that just about the time we got our first chicks and first turkeys, I had interviewed Dr. Virginia Livingston Wheeler. And so I insisted and persisted, and we did derive that vaccine through her, and every little chick and every new turkey, they were all vaccinated, and if they reproduce, or the new ones, they of course get vaccinated also. We have excellent fowl because we let them run outside in the fields instead of keeping them in boxes as they're they're raised elsewhere, and so the eggs are good too. But I just want everyone here to know that since I became aware of that information, I wasn't about to allow anyone in this community to be partaking of contaminated fowl. And one of the things that was very interesting to me was Dr. Virginia was speaking to me both personally and on the show about just how the fact that all chicken is cancerous. And she told me this story about someone that I have never forgotten, but she was at speaking with someone over the phone, or someone who was holding a cocktail party, and this person loved chicken livers. And so she told her that cancer is heavily concentrated in the liver of the chicken, and warned her against eating it. And she looked at me and she said, this person said, well, I love chicken livers and I'm going to go ahead and eat them. Ate them, and it was a matter of months after she had been warned by Virginia that she was dead of cancer. So this is nothing to fool around with.
Right. I mean, this is a topic that's intimately connected to this whole problem. Now, the theory is that the oncogenes, I mean, we spent $65 billion. We have nothing to show for it that I can see much except for AIDS, and a theory that doesn't make much sense. And the theory is this, that as if we're all supposed to jump up and say hooray, because they say now included within each human being are certain genes that could arise that give cause to cancer. Well, I mean, you don't have to be a real genius to figure out that each of us probably carries that innate potential to cause cancer in us, because otherwise, where is it coming from? Because if it's not infectious, well, then it's arising from something. It's arising from within. So in the fact, the oncogene theory, there is not one oncogene that exists in nature. All the oncogenes that exist in the wild are in fact what we call proto-oncogenes, or pre-cancer. And there must be an internal rearrangement of your genetic makeup to give rise to the oncogene before it can cause a cancer. The same thing as in chickens. This is an interesting experiment. They took the cancer gene of chickens, they put it in its final form, and they transduce it with a virus of this nature, a retrovirus, into 1,000 chickens. All of those chickens get it, they all die. They take the part that actually existed in nature, in the wild, before they started all their mass breeding programs. If they transduce the proto-oncogene, the true part that existed before you might have mucked around with it, into 1,000 chickens, none of them die and none of them get a cancer. The same thing is true in humans. If you look at the so-called cancer gene theory of humans, they say, well, this gene looks a lot like that gene, and so it's probably, the cause. It's like saying, well, sickle cell hemoglobin looks a lot like normal hemoglobin, so why wouldn't you want to change yours for sickle? Well, it's because it doesn't work the same. It gives you a disease. And so if you look at man in the oncogene, proto-oncogene theory, there is not one of these cancer genes that actually exists in nature. So you're then still left saying, in a sense, which came first, the chicken or the egg, or in this case, which came first, the chicken or the cancer. So you don't actually have a clear-cut answer. I think that a great number of these oncogenes, which exist in nature, obviously exist in their pre-cancer form, because if they existed in the cancer form, you would get a cancer and die. And so in the wild, cancer is outbreeding. Animals that have dominant genes for cancer get cancer and die. If your chickens live long enough and they have the cancer genes in them, they're all going to get leukemia and die, provided you don't eat them first. Or in this case, we have good chickens. So, but it's back to the oncogene theory. They're now pushing this as saying, well, this is going to satisfy all of our explanations for cancer causation. And still in my own thinking, having read thousands and thousands of papers on both retroviruses and the cancer and the relationship, it's not clear to me by any means that retroviruses or the proto-oncogenes actually are the cause of cancers. It's still unclear to me.
For all you do know, you've made some very dire predictions at the end of your film. And I'm wondering if you'd restate them here.
Right, we predicted, well, let me turn to the blackboard again in a second. We predicted that this virus has the capability of destroying mankind. If you look at the numbers of people in the United States that are infected, say 2 to 4 million, if it doubles every year, it can go to 4, 8, 16, 32, 64, 128, or over here 256 in 6 years if it doubles every year. Since 1978, the number of AIDS people infected in the United States and the number of cases have been doubling every year. So you can predict that perhaps in 6 years you could have everyone infected, and that would be before anyone showed evidence of disease. And that's with the AIDS virus alone. You also have to worry about its relatives, HTLV1 and HTLV2, the leukemia viruses. So we think that everybody could be infected before anybody knew. The question is, how could that infection occur? You know, you say, well, I'm not a homosexual, I'm not in the 4-H club, I'm not a hemophiliac, you know, I'm not an IV drug abuser, I'm not from Haiti. So how am I going to get infected? Well, there could be the fact that if you look in Africa, for instance, across the AIDS belt, that there is a critical mass of infection at which time, no matter what group, so-called risk group you're in, and this whole thing of a risk group is really, in a sense, peculiar, but no matter what group you're in, once you reach a certain number of infection, then everyone will get infected by whatever mechanism infection is occurring. And still to date, can anyone, any doctor, produce for us the actual mechanism how this virus infects another human being? I certainly haven't seen it published, and I don't know about you guys. Have you seen any explanation of that anywhere? No, you know, nothing. And yet they tell us they know all about it. It's entirely possible. Everybody wants to know about mosquitoes. So,
right, can you get infected?
Yeah, the mosquitoes. Could this, you know, at first, if you go back, read back in 1981, 82, 83, you know, they said, you can take 100 pints of blood and there's no problem. Then they said, well, of course, whoops, 60 pints, no problem. Whoops, 1 pint, no problem. Whoops, blood, stick, no problem. Then they went to needle stick, no blood, no problem. And then they said, well, needle stick, period, no blood, you may have trouble. So the history of what they're telling you about this virus being transferred between people by vectors, particularly in this case, a vector is a needle, was totally incorrect and, of course, dangerous. Now, if you look at AIDS, all of its near relatives, which are caprine arthritis encephalitis virus, equine infectious anemia virus, bovine leukemia virus, Visna virus, all of these are transferred by vectors such as mosquitoes, or other blood-sucking insects such as tabanid or horse flies. So, you say to yourself, why is it that this virus, which acts like these others in every other way, a lot of ways, won't be transferred by mosquitoes? There's a very simple experiment that has yet to be done. We take a few of these AIDS researchers. We have a box here filled with mosquitoes. We let them feed on a bunch of blood that has AIDS in it. And then we let them bite the researchers. And we see, no, I'm serious. And we see how many of them get AIDS. Now, if you really believe that this virus is not transferred that way, why are you unafraid to do that experiment? But they haven't done it. In fact, they haven't even done it with chimps or anything else. Additionally, if you believe that this little mosquito here, which, you know, is sucking up blood, if he doesn't transfer the virus during his blood transfusion to you, why is it that we, as doctors, are transferring the virus during our blood transfusions? Why don't we employ the same mechanism that the mosquito does to filter this virus out arbitrarily? So, now if you look in Africa, they say, well, you know, if AIDS was vector-borne, everybody in Africa would have it. And of course, what appears to us is in Africa, Everybody does have it. So, if you look at the incidence of AIDS in Africa, and you compare it to age, you say, well, it looks like this. And of course, this is the AIDS incidence curve for AIDS, and this is the AIDS incidence curve for another disease known as malaria. So you can see that the AIDS incidence curve for malaria, which we know is vector-borne, and AIDS are virtually identical. So the burden of proof to say that this virus is not transferred by vectors lies in a very simple experiment that has yet to be done. And that is, let's see, take this virus, let the mosquitoes feed on it, let them bite some of these researchers, and see if any of them get AIDS. So,
I just got some things I'd like John to run over quickly, such as relationship between pesticides and AIDS, pharmaceutical companies foisting their pesticides on the third world, and then AIDS infecting the brain, AIDS dementia. What do you have to say about those subjects?
If you look at any standard text on pesticides, you will see that they are in the most severe form nerve toxins. They can bring about a number of the listed symptoms in various definitions of AIDS. We know that the very worst pesticides are dumped into the third world. A good example is Brazil. We talk about Brazil as being a seething country of underreported AIDS cases. There is a policy of Western corporations sending the most dangerous pesticides to be used into Brazil, and they send the ingredients separately to get around various regulations, and then the ingredients are formulated in various factories in Brazil and then are used out in the fields. Now, what you have to understand about these pesticides is, for example, parathion was a chemical warfare weapon of the Nazis during the Second World War. Slightly altered becomes a pesticide. Very dangerous pesticide, possibly 60 times more dangerous than DDT, which has been banned in the United States, but is used extensively in the third world. Without labels, or if there are warnings, they're written in English, which most people in the third world don't bother to read. They can't understand them. And they are handed around very easily to people. Sometimes they are mixed up in containers that are not covered. In Brazil, we have some very good reporters that estimated that there are 8,000 backyard whiskey still operations going on where people in Brazil, to make a few bucks, are mixing together these various formulations for pesticides in Coke bottles, in anything, you see, and handing them out to people to go out in the fields and use these. This can produce a profound immune suppression in the human body, especially coming through the route of as being nerve toxins, and it causing tremendous and fairly rapid weight loss, known as the wasting syndrome, under the list of AIDS symptoms. So when you have pesticides like these and pharmaceuticals, which are also quite dangerous, the most dangerous of which are also available in certain third world countries, certain wide use of the most dangerous, for example, antibiotics like chloramphenicol, you are opening the door to a large amount of immune suppression plus opportunistic infections among people. And although I haven't been able to flesh out the full theory in AIDS Inc. here, I'll just make this remark, and that is the basic pattern of AIDS is immune suppression plus opportunistic infection. One of my points is that you can, with chemicals, cause immune suppression plus opportunistic infection. You can bring about the same identical pattern of AIDS by use of chemicals. And in fact, that this has been done. Where I think we have to really think about this in political terms here is AIDS is going to become a smokescreen for whatever is happening in the third world. I could sit here for 3 hours and tell you about rampant disease of different kinds in the third world, of causing tremendous problems. 14 million deaths from measles since 1978. You're not reading about that. 14 million deaths from measles since 1978. Want to talk about an epidemic? Talk about that. Now, when you load into a country a tremendous quantity of what we call endemic disease, tropical disease, accelerating tropical disease, like malaria, huge use of pesticides, dumping drugs that are too dangerous to use in this country into areas like that, you are mixing together some incredibly explosive factors that can cause things that you never dreamed of. Add one more factor to that, which is chronic generation to generation malnutrition and starvation, which happens to be the biggest cause of immune suppression in the world, which can depress T-cell numbers, which can cause macrophage defects, which can cause all the manifestations that we have come to think of as AIDS. And you have to begin to wonder, does anybody have a motive for trying to mask off some of these horrible things that are happening in the third world and erect a 50-foot sign across Brazil that says AIDS, when in fact what is happening in many third world countries is a political situation in which those countries can manufacture and are manufacturing enough food to feed their people if they would divert but approximately 5% of what they are now manufacturing instead of exporting it back to the industrial world. But the agriculture of the third world is by and large controlled by multinationals. And these multinationals care as much about that situation as you and I care about, what time it is right now. They don't care. So you have to say, well, what is the motivation here? And what you're going to see, I predict, in the future is you're going to see that all manner of disease that develops from things like tearing down the rainforests, which produces tremendous upsurges in quantities of disease like river blindness, malaria, because the malaria mosquitoes come down from the top of the canopy of leaves when you tear down a rainforest, and at night they enter people's houses. You're not going to hear about all of this kind of stuff. You're not going to hear about the fact that there are very nasty parasites that breed on the cattle that are now grazing on the land where the rainforests used to be, that the multinationals have taken over, stripped away, and set up as grazing pastures. Instead, what you're going to hear about is the devastation of AIDS, period. And I'm here to tell you that after a year and a half of researching this, and there's a chapter in AIDS Inc. on the situation in Uganda, that just about any vector of disease, chemical destruction, pesticide use, the whole mishmash, and throw in malnutrition that brings about future generations of people with smaller heads and smaller frames, because we're talking generations of malnutrition. And you say, wait a minute, what's going on here? If you now look at the definition of AIDS in the third world, and I suggest you read the World Health Organization definition of AIDS in the third world, I suggest you read in the October 24, 1987 Lancet magazine, a medical journal, an article called Clinical Case Definition of AIDS in Africa. You will find that the three major symptoms now in the new definition of AIDS in the third world are the following: chronic diarrhea, 10% weight loss or more, and chronic fever. Well, diarrhea has been the third or second largest cause of death in the world forever. It's called dehydration. And if you take those three symptoms together that I just named, you have what happens to someone who is chronically malnourished over a long period of time. We are now presenting a definition of AIDS in the third world as being identical to starvation and malnutrition, so that we're going to see a tremendous inflation of case statistics in the next 5 to 10 years of AIDS statistics based on the fact that we have continuing and growing starvation and malnutrition all over the world. Because doctors, using this definition, are not supposed to go to laboratories and make the careful tests of blood. They don't have the laboratories, so they eyeball the diagnosis from the definition. That's why the definition is so vague. Weight loss, chronic fever, chronic diarrhea, AIDS. One more, let me pile on top of that. Politically, this opens the door for what I think is one of the most threatening possibilities. I don't say it's going to happen tomorrow, because it isn't. I don't think it's going to happen in the next 5 years. But 10 years, 15, 20 down the line, we are talking about a form of political repression that needs no ideology. Perfect if you want to be a repressor. You don't have to talk communism, you don't have to talk capitalism, you don't have to talk regimes and divine right to rule or any of this stuff that's been used to suppress people from day one. All you have to do is work out a deal with medical authorities from, say, here to go into the third world, somewhere where they would like to establish more firm political control, especially if they got a lot of minerals there under the ground, and they declare medical states of emergency. See, the authorities have decided that it is no longer safe for this population to exist under the present regime. We're going to have to institute very firm measures here. People are not allowed to congregate in groups of more than 3 because of contagion, blah, blah, blah. And this guy, who is a political dissident anyway, we find out he's positive for the virus, throw him in jail, and we're going to take over. But we're not going to take over on any program or platform. We're going to take over with the armaments of the modern priesthood, which is white coats and science. That's what more and more people in this world are now worshiping. Because worship is defined as you accept on authority alone, without inspection, without the power of your mind, somebody else's opinion. That is the attitude of the public towards science. And so if doctors move into a country and say, it's a medical emergency, we're now going to institute a more dictatorial rule here, we're going to take over, we're going to set up detention camps, we're going to do this and that. On the surface you can say, well, it's all for health, you see. I mean, naturally. Because it is a problem. We have tremendous health problems. But I urge you to look behind the motivation for that. The motivation is from, let's call it, we've heard something now about the Soviet side, and I agree that the Soviet side and the American side, I mean, flip a coin. But look at the multinationals. Look at their interests. Their interests are in money, money, and more money, and not people. If you, as the chairman of the board of a multinational corporation, thought that it would be more expedient for you to go into a third world country and develop their mineral resources by getting rid of an excess population who could not only be unproductive in terms of, you know, you only need so much labor, especially as you develop more machines, but could present a revolutionary problem to you, because they're all starving to death and they perceive the only answer is to overthrow the government, why not move in with medical states of emergency? No political ideas are necessary. See, it's all for your own good. That's all you have to know. And the scientists know everything, and they've got all the high-tech equipment, and you know nothing. In this country, there is the nub of that idea. And you might be surprised to see how fast it could spread if fear became an uppermost plank in a political platform in this country. And so I think everyone needs to adjust their viewpoint here, not simply the biological elements involved here, but some of the other motivations. And you begin to see how people who are motivated in a very nasty, nasty, nasty way, people who, for example, might like to see the world as more or less a country club, where you've got nice rolling hills and not very many people, you see. Let's clear out the riffraff so that I can look down from my house across, 3 states, and all I can see is some very nice people that make boots and, they carve charming, quaint antiques that I can buy and so forth. This world is too crowded. And people like this, and there are such people that exist that have power, they think in terms of strategies. And this is a strategy that I believe they are thinking of.
And they think in terms of ownership. They own you.
That's right. They own you.
Right, I mean, you can envision the difference between, say, an invading army marching down the streets of your country bearing arms. What kind of resistance would an invading army meet as compared to invading doctors who come in and say, here, we're going to fix you. Just take this shot. I mean, doctors are accepted. They're trusted. Whereas an invading army would meet automatic resistance. So, and then of course the doctor says, well, we have a state of a medical emergency here and we're going to institute all these controls.
I'll tell you a little secret somebody told me. Good source, I think. Certain people at the Centers for Disease Control go up to Langley, Virginia to get trained by the CIA. It's kind of an odd mix, you might say, you know. These are the people that are supposed to be statusizing disease in the country and Centers for Disease Control. And there are certain people that go up and they become trained by the CIA. The source revealed to me that there are certain files at the CDC that are marked top secret. And it's strange, but they noticed that the people that seem to have access to those files are the people that happen to have gone up to Langley to get the training from the CIA. And information comes into the CDC from all over the world, and it is categorized. And I've been told this by a spokesperson for Health and Human Services. This is a public fact. There is confidential material, there is open material, there is top secret material in files at the CDC, and they're all different. And you begin to say, well, what's so top secret about disease? I mean, you know, disease is something we want to cure, right? What's the concealment factor here? And so the trouble is rampant.
Okay, let's take up the cure.
Okay, let's talk about how we think these diseases can actually be fixed. In 1925, a guy named Raymond Roy Rife created what I think is perhaps the way that this disease can be fixed. It's very simple in principle. What Raymond Rife said was the following: if this was a crystal glass and you irradiated it with the proper audio tone, you can shatter the glass, but if you were holding it, your hand doesn't fall apart. Now viruses, AIDS virus, many other viruses, are really crystalline in nature. And so what Rife said was that human beings that were infected with these viruses could be irradiated with a radio tone, a radio wave, that would vibrate the virus, shatter the virus, and the human body would not be affected. And he says, he said that not only was that possible, but that he did that in his experiments and was curing virus diseases and cancer with this technique in the early 30s, late 20s. He said it would be a fix for not only viral and bacterial diseases, but for certain types of cancer. Now, I don't know if he really cured cancer or if he really cured any virus or not, but there's a certain appeal to this technique that I think might work. In 1931 or so, a guy from India named Raman won a Nobel Prize for opening up a field of physics called Raman spectroscopy. And what Raman showed was that molecules do absorb energy and then re-emit energy of different wavelengths depending upon the molecule and what you put into it. So it shows that maybe Rife was not just whistling Dixie, he was actually onto something. Of course, today doctors say, no, that's just not possible, you know, that's insanity. And yet you ask them, how do they kill viruses in their laboratory when they produce these things? They blow them out their hoods, they collect them on electrostatic plates, and then ultraviolet lights kill the virus. And well, ultraviolet is nothing more than electromagnetic radiation of a certain type, a certain frequency. So it is possible to kill viruses. It's undoubtedly true. Now recently some studies have been done into this field in Baylor University, which took blood that was contaminated with AIDS virus, cytomegalovirus, and some other herpes-like virus, took the blood, irradiated it with argon or xenon, I can't remember which, but one of those two laser lights, and showed that they killed the virus without killing the blood cells. So this is more than just potential theory. I think that the actual cure for this disease, and perhaps a lot of other viral diseases of humans, lies in either the redevelopment or the reconstruction or in the production of instruments that will, in a sense, tune to these organisms and destroy them without destroying the human.
Is anybody doing it?
I don't think there's anybody doing it except for people at Baylor who are working on the argon and xenon lasers that they've used on certain viruses. There are very few people who, again, believe that this is even a theoretical possibility.
Sounds to me like you three are just kind of the guys that ought to get into that very experimentation.
There's quite a few implications for what Bob's talking about here. I have come across in interviewing people who in the last year are trying these alternative methods, you know. The basic fact that most of the time you're dealing with I think you call him the garage tinkerer. This is the character who doesn't particularly buy what's, you know, the history of science. He says, well, maybe there's a better way to do it. And people still, unfortunately, labor under the delusion that our health system is made up of such people, when in fact what's been slowly happening, like some creeping fungus for the last 50 or 60 years, is the growth of these gigantic agencies, federal agencies that are federally funded, and huge university research departments where this kind of research does not happen. See, it's been obliterated from the landscape. So instead of having a Royal Rife who invented a magnificently interesting microscope in the 1920s, became famous even in orthodox medical circles, eventually was wiped out, you now don't even hear about the person. There are many, many people, I want to tell you, that are working on this independently, with no funding, just on their own hook. And the I call them the electromagnetic people. They're all over the place. They've got different kinds of machines, you know. And sure, there's some people hyping, and there's some con men at work, but that's not the point, really. AMA would like you to believe that it's all a con. No, there are people working on interesting things to boot. And I've come across some people that say, you know, I had AIDS 5 years ago and I don't have it anymore. I feel good. And I have to tell you, my personal opinion is, and one of my bottom lines here is that emotionally I feel that the person himself, regardless of what you say, the person himself can make a difference. The person's attitude toward themselves, the kind of thing you cannot factor into any sort of, you know, equation, is a very big deal. And among the people I talk to that call themselves survivors, they emphasize that fact. There was one curiously interesting thing. None of them had followed a protocol from any doctor, orthodox, allopathic, holistic, whatever, for very long. They would move around and they would do, as you said, they would experiment on themselves. They took charge of the situation and they said, I'm not gonna be one of these people that does this. I'm going to work on this, you see. And here they are, and they worked on a lot of things.
On these notes of hope, I'd like to thank you, Dr. Robert Strecker, Mr. John Rappoport, and Dr. Alan Cantwell for being our guests at Summit University Forum and the flame of freedom that speaks.
Thank you.
You've been watching Summit University Forum with Elizabeth Clare Potter. Summit University Forum, where the flame of freedom speaks. Elizabeth Clare Potter, noted author, lecturer, and educator, interviews the special people who provide the missing dimension to the news that affects you. Courageous men and women from all over the world who have taken a stand for truth and justice and become spokesmen for the flame of freedom. If you have any questions about today's program, call 406-222-8300 or write Summit University, Department V200, Box A, Livingston, Montana, 59047, 1390.
I will have the glory of my immaculate, divinely conceived Son, manifesting within me. I am freedom and I am determined to be freedom. I am the flame